Antibiotics are some of the most important drugs that we use in medicine and in pediatrics in particular, but many students find them difficult to learn. So I would like to share my approach to learning the antibiotics. I like to start by organizing antibiotics by mechanism, whether they're cell wall inhibitors or protein and DNA inhibitors. And we'll talk about the difference between those two types and why it's useful to think about them in that way. I think that less is more. There's a lot to know about antibiotics, and you will build your knowledge over time. But when you're just starting out, it helps to start with just a couple facts about each antibiotic. The two facts that I think are useful to know, which is knowing one side effect and one indication for each antibiotic. It's great to know more, but that's a good place to start. There are four main categories of antibiotics. These include antibacterials, antifungals, antivirals, and antiparasitic drugs. We're going to focus on antibacterials exclusively. These other drugs are very important but have slightly different mechanisms of action, and we're not going to cover them at the moment. I also want to talk about the fact that every drug in medicine, or for the most part, almost every drug in medicine, has both a brand name and a generic name. In school, we typically learn the generic name, but we often use the brand name in clinical settings such as the office, in the hospital. So I will give both the generic name and the brand name, but during these talks I'm going to try and use the generic name as much as possible, but it is very valuable for you to learn both names. So I like to split antibiotics up into cell wall inhibitors and protein and DNA inhibitors, and we typically will include other mechanisms like folic acid inhibitors into that same category, sometimes cell membrane inhibitors. So I split it up into cell wall inhibitors and other mechanisms. What type of bacteria are cell wall inhibitors effective against? Pretty straightforward. You have to have a cell wall. You have to have a cell wall, and it has to be easy to access the cell wall. So bacteria that are obligate intracellular, which means they have to be inside human cells, typically cell wall inhibitors are not so effective against them. So what kind of bacteria do we have here? Mycoplasma does not have a cell wall, so cell wall inhibitors are typically ineffective against mycoplasma. And then some bacteria that are obligate intracellular include chlamydia, rickettsia, which is the cause of Rocky Mountain spotted fever, and P. jirovecii, Pneumocystis jirovecii. That's actually a fungus that we slipped in here, but again, obligate intracellular, our cell wall inhibitors are not effective against this. Legionella is a facultative intracellular bacteria, which means it spends sometimes inside the human cells, sometimes outside human cells, but in general, cell wall inhibitors are also not effective against Legionella. Now, protein and DNA inhibitors are still effective against many bacteria with a cell wall. So they are good for many types of bacteria. Cell wall inhibitors are a little bit more particular. You have to have the cell wall. And I sometimes split this up by saying that cell wall inhibitors are good for normal bacteria and protein and DNA inhibitors are a little bit more accepting. You can use them for the weird, unusual bacteria that don't have a cell wall, that are obligate intracellular bacteria. They have a broader spectrum of action. And they'll still be good against bacteria with the cell wall, as I mentioned. So type of bacteria is one way to separate out these two classes of antibiotics, and then type of resistance is a second way of separating them out. For the cell wall inhibitors, we have many of the penicillins have a mechanism of resistance called beta-lactamases. If we look at the structure of a penicillin, we see these two chains, two circular structures, two rings, and one of the rings we see is the beta-lactam ring, which I'm highlighting right here. Some bacteria produce a beta-lactamase that destroys this ring, and so that is one of the mechanisms of resistance. We can overcome this by adding a second molecule called a beta-lactamase inhibitor, and we'll talk about that more in just a little bit. A second mechanism of resistance in bacteria that produces a cell wall is called a penicillin binding protein. Bacteria with cell walls produce these set of proteins that act to cross-link the cell walls together. Now, they weren't produced to bind to penicillin. They were produced to cross-link the cell walls, but that is a site where penicillin can bind. And so one mechanism of resistance is a change in these penicillin binding proteins that prevents penicillin from binding effectively. When we get over to the protein and DNA inhibitors, we see a different set of mechanisms of resistance. So one is increased efflux. The bacterial cells will pump the antibiotic out so that they no longer work. We may also see decreased binding, similar to the decreased binding in penicillin binding proteins, but obviously a different set of proteins, where we may see decreased permeability. The antibiotic just can't get inside the cell membrane of the bacteria. So to summarize, it's useful to split antibiotics up into cell wall inhibitors and other mechanisms, including protein and DNA inhibitors, because the two types of antibiotics are used to treat different types of bacteria and have different patterns of resistance. The quick way I think about it is that cell wall inhibitors treat normal bacteria that have a cell wall and that spend some time outside human cells. Protein and DNA inhibitors can still treat bacteria with a cell wall, but they also are good at treating some of these weird, unusual bacteria that don't have a cell wall or are obligate intracellular or potentially facultative intracellular bacteria. And the pattern of resistance is different. With cell wall inhibitors, we can see changes in the penicillin binding proteins, or we can see production of a beta-lactamase to disrupt the beta-lactam ring of penicillins. And with protein and DNA inhibitors, we'll see increased efflux, decreased binding, or decreased permeability of the cell membrane to the antibiotics.